All right, great. Why don't we get started? It's really a pleasure to welcome Dr. Razelle Kurzrock for this Cancer Center invited seminar. Dr. Kurzrock is currently the Chief Medical Officer with the WIN Consortium for Personalized Cancer Therapy. I got to tell you, earlier today we received her CV. It was 199 pages, so I could spend the next week going through it, but I won't. What I'm going to do is give you some of the highlights, and especially for the students or others who have not met Dr. Kurzrock, she's been incredibly prolific. There have been few CVs in my career that I've looked at that have had as much accomplishments over the last couple of decades. So she lists 865 publications as of August 2021. We were talking about Twitter earlier. She is the #2 female medical oncologist on Twitter worldwide. So that's, I was trying to remember or figure out who #1 was, and I don't know if it was Dr. Powell or somebody else, but Dr. Kurzrock's H-index is 129, and she has almost 70,000 citations. And for those of you who submit meeting abstracts, she has more than 1,500 of those. So Dr. Kurzrock earned her MD degree from University of Toronto, did an internship at Sinai in Toronto, and then a residency in medicine at Tulane. Followed by a fellowship in hematology oncology at MD Anderson Cancer Center. Most recently, before joining the WIN Consortium, she was Distinguished Professor of Medicine and Leader of Experimental Therapeutics, Associate Director of Clinical Science at the UCSD Moores Cancer Center. And while there, she was also chief of hematology oncology for 5 years. Before moving to UCSD, she had spent most of her career at MD Anderson, where she was, at the time she left, the chair of the Department of Investigational Cancer Therapeutics. She held many leadership roles at MD Anderson. And was co-director of the Center for Targeted Therapy there. I saw on her CV she also spent a year long ago at the Wistar Institute in Philadelphia in the laboratory of Dr. Carlo Croce. Dr. Kurzrock is currently senior editor with Molecular Cancer Therapeutics, and also the reviews editor for that AACR journal. She serves on the executive committee of the NCCN Board of Directors. She's a member of the Scientific Advisory Board of SWOG and the chair of the SWOG Early Therapeutics Committee. She serves on a bunch of external advisory boards at including Yale's Cancer Center and University of Miami's Sylvester Cancer Center, many scientific review committees nationally, AACR, PANCAN. She chairs the Scientific Advisory Board of Actuate, and serves on many other boards, service, and leadership roles. Like 20 pages of her CV are just about that. So, you know, she's an amazing person, and I think we're honored to have her with us here today. In 2021, Dr. Kurzrock was the San Diego Business Journal Most Influential Women in Technology, has had many other awards. She has held funding on 92 grants, including 10 in 2021 that were active, 14 patents on liposomal curcumin alone, has served as PI or co-PI on 107 clinical protocols. And, you know, looking over her CV, the early part of her career, she was really in the area of malignant hematology. And she was quite active. You know, she published on in Philadelphia chromosome positive ALL, and, you know, various JCO and New England Journal papers and other manuscripts. And then about 15 years ago, she turned her focus, about 15 years ago, more in the area of tumor-focused research. And for some of the you who are interested in clinical trials, I mean, she led the phase 1 experimental therapeutics program at MD Anderson. And for anyone interested in doing early phase trials, in 2009 in the JCO, she published a paper called on Project Zero Delay, which anybody who's interested in clinical trials, this is worth a read in terms of how institutions could do a better job of opening up clinical trials, and it recounts some of the accomplishments at MD Anderson. In the last 10 years or so, she's worked on targeted therapeutics. Dr. Barry, can you please mute your Zoom? So if you look at sort of the various types of clinical trials throughout the years, she's worked on so many targets that are important in cancer and across various pathways. In recent years, she's advocated for a reclassification of cancer and universal genomic testing and profiling as some of her recommendations. You know, she's considered a premier expert in drug development, and it's really, really great to have her with us today, where she will speak about the next generation precision genomics and immunotherapy. So welcome.
Well, thank you for that very nice introduction, and you really covered a lot of ground in my history, so I really appreciate the attention. So I'm really excited to be speaking to everybody. First, let me confirm that everybody can hear me and see my slides. Yes?
Yes.
So what I am going to talk about is next generation precision genomics and immunotherapy. I believe that precision medicine is really transforming cancer. It will ultimately transform all of medicine, but for the most part, I'm going to stick to cancer here. And these are my disclosures. And, you know, as was mentioned, my most recent endeavor was at the University of California, San Diego, where I held several leadership positions. But the one closest to my heart was that I founded and directed the Center for Personalized Cancer Therapy. And before that, I founded and chaired the Department of Investigational Cancer Therapeutics, which is the phase 1 department, which within 5 years really became, I believe, the largest of its type, probably in the world. And we were putting 1,300 patients on therapeutic trials a year. And it's these two experiences that really informed my viewpoint about precision medicine, especially initially the experience at MD Anderson, where for phase 1 trials, we were treating patients that by definition had failed all FDA-approved therapies. They had a terminal disease with no treatment. And we began around 2007 incorporating very primitive genomics by today's standard, but we began to be able to see a glimpse of what was possible. And of course, the field has moved tremendously since that period of time. So I want to tell you the take-home points. The take-home points is to give the right drugs to the right patients at the right time. And that breaks down to, at the genomic level, every metastatic tumor is unique and it's complicated, akin to malignant snowflakes. I lived in Houston, I live now in San Diego, but I actually grew up in Canada. So I know a little bit about snowflakes. And every one of those snowflakes has a unique and complex crystal structure, and I believe metastatic tumors are similar. And therefore, metastatic disease requires customized, individualized combination treatments, not single agents, and for the most part. There are exceptions. The pillars of precision medicine are genomics and immunotherapy. They are often considered separate areas, but I believe that they're a couple and they're married to each other. And a lot of targeted therapeutics is best suited for simple disease. Immunotherapy, however, is best suited for genomically complex disease. So what has happened in the last couple of decades is absolutely breathtaking. And I'll tell you that in the 1990s, it was conventional wisdom that we would never be able to sequence the human genome because it was too complicated. Never. But by 2003, never was over. And the human genome had been sequenced. It took 13 years. And amazingly, today we can sequence within a day. And it took $2.7 billion to sequence the first human genome. By 2007, when we started doing genomics at MD Anderson, we were down to 100 million a genome. So that was still a little bit high for insurance to cover or for my research grants. Even if I was well funded. And so we had very primitive genomics, but now we can sequence a genome for $1,000. And, so a lot of people complain about the cost of sequencing, but really this is breathtaking progress, I think almost unparalleled in human history. And it's really enabled what the transformation that is happening in oncology. So what's happening to the evolution of clinical trial design? As an example, I'm going to use lung cancer as an example. We thought it was one disease. Now we began to understand that it's multiple different subsets defined at a molecular level, KRAS, EGFR, BRAF, and so forth. And then the next step is that we began to understand that different cancer types defined like organ of origin, breast cancer versus colon cancer versus lung cancer, could have the same genomic alteration, and that we could target them in the same way. And this meant that we began to move from large randomized trials of unselected patients where only a few, very small number of patients in each arm would respond, to highly selected populations selected on a genomic level where and with very specific therapies targeted to that gene aberration, where we would expect maybe even the majority of patients to respond. And this is the redesigning of cancer clinical trials stage 1. As we talk, I'm going to walk you through stage 2 as well. But this gave birth to a new era, and that is tissue-agnostic approvals driven by genomics. And I just want to say that around 2007, we put into the MD Anderson IRB what I believe may have been the first tumor-agnostic study. It was a study for the mTOR inhibitor temsirolimus for patients that had PI3 kinase pathway alterations. And that was really considered almost insane at that time point. But it's just amazing how fast this has moved. So on May 23rd, 2017, and I think the date is important because this was really a breakthrough, the FDA approved for the first time a therapy that was tumor agnostic and based on a genomic defect. And there's three things that are really important about these approval. The first two, most people know, and then the third is, I think, the most important, but most people don't realize it. The first is that it was a tissue agnostic approval, so it's really approved for all solid tumors. And the second was that it was approval based on a genomic marker. And then the third, and this is really important, it was approval based on retrospective, in part on retrospective real world data. So the FDA opened the doors for real world data without a trial. And since then, there's been approvals like palbociclib for men with breast cancer that are entirely on real world data. So there is really some incredible implications of that. But that was the beginning, not the end. So in 2018, we had larotrectinib, which was approved for NTRK fusion solid tumors, and then in 2019, another NTRK inhibitor approved for solid tumors. And these were approved across the board, tissue agnostic, and the response rates were anywhere from 50 to 80%, depending on the study. And then in 2020, we had pembrolizumab, checkpoint inhibitor, approved another tissue agnostic approval for patients with high tumor mutational burden. But there's still even more in that direction. And belzutifan was just recently, a few weeks ago, approved for patients with VHL disease, regardless of the type of tumor that they have. And then finally, we have multiple checkpoint inhibitors. So we now have another checkpoint inhibitor, again approved about 4 weeks ago for mismatch repair defect solid tumors. So this is a real paradigm now with multiple approvals. And so that brings me to where I want to walk back a little bit and talk about genomics and immunotherapy and how they may be related to each other. So I think genomics and immunotherapy are the pillars of precision or personalized medicine. And a lot of people feel that they're separate. I mentioned this before, but I think they're a couple and they're married to each other. And what do I mean by that? So it turns out that the immune system responds to the mutanome. So the mutations are processed to neoantigens, and it is the response to those neoantigens that drives the responses to therapy. And therefore, not surprisingly, genomic aberrations can predict response to immunotherapy. So just a few examples: high mutational burden, PD-L1 amplification, mismatch repair gene defects, and defects in genes such as chromatin remodeling genes can predict in a very powerful way responses to immunotherapy. And then, I'm going to give you the example of tumor mutational burden, which I know has generated some controversy, but my experience in our publications and my experience in the clinic is that it is a very, very powerful predictor of immunotherapy response. So at data from UCSD at a low mutational burden, 4% of patients respond to checkpoint blockade, intermediate about 26%, 45% rate for high mutational burden, which we think is 20 mutations per megabase or higher, and 67% response rate for very high mutational burden. And in some cases, these are complete remissions that are durable. And so the more mutated the tumor, the better the response to immunotherapy. And I want to make two comments about that. The first comment is, this is again how science changes everything. If we go back just a few years ago in our molecular tumor board, and we would see these horribly mutated tumors with all these mutations, and this was a really messed up cancer, and we would sort of hang our heads and feel bad for the patient, because like, what are you ever going to be able to do with a cancer that is just chock full of mutations? And then just a few years later with immunotherapy, it turns out these are the best tumors. We get so excited when we see a very high mutational burden tumor, because we think that that patient has a really good chance, or a reasonable chance, of a really good response, and maybe even a complete remission that is durable. And so why is that? Immunotherapy, checkpoint blockades, reawakens the immune system. The tumor, in order to survive, has put the immune system to sleep. Checkpoint blockades allows us to reawaken that immune system, but that reawakened immune system has to recognize the tumor. And as you might expect, if there's only one or two mutations, that tumor may be too similar to a normal cell for the immune system to recognize it. But if the tumor is just chock full of mutations, you've rolled the dice in favor of the immune system, once reawakened, to be able to look at that tumor cell and says, that doesn't belong here, I'm getting rid of it. So really with science, the situation really changes completely for groups of patients. And so we established a rare tumor clinic, and this is an example, which is a precision medicine clinic. And I want to say that rare tumors is a special interest of mine. Individually, they're rare, but altogether they make up about 25% of the cancer burden in the United States. And they're a very big unmet need. And so we really did this for all patients, but we had a special emphasis on rare tumors. Patients came into clinic, they signed a master protocol consent, and then we did all sorts of biomarkers, next generation sequencing, liquid biopsies, protein analysis, immune signature analysis, and so forth. And this was our molecular tumor board. We initiated at the end of December 2012. It had both an electronic form and a weekly face-to-face. All in all, we did about 21,000 clinical grade molecular profiling. And one of the keys to bringing this to patients was that we had an umbrella protocol for the cancer center. So if a cancer patient walked in, they could be consented to the umbrella protocol. They could automatically go on study. We could do translational research with their material, and we could treat them. And the molecular tumor board, very importantly, was multidisciplinary. It was attended by multiple different physician types, from medical oncologists to surgical oncologists to gyne oncologists, radiation therapists, radiologists, pathologists, but also by our basic scientists and by our bioinformaticians. So really brought together multiple disciplines. And the idea, this was not a research exercise. This was an exercise in trying to figure out the best therapy for patients and how to make that happen for patients. So I'm going to give you some examples of like patients that I treated, just to illustrate you know, what can happen. So this is a 55-year-old man that was a patient of mine. He had metastatic basal cell carcinoma, came to see me in October 2015. He had failed multiple therapies, including the FDA-approved therapies such as vismodegib and sunitinib, which are hedgehog inhibitors, because these patients have hedgehog defects. And he had brain, bone, and lung metastases, had failed 6 therapies, was not in very good shape. And, you know, so what would you do most likely next for this patient? So I think, you know, maybe send to hospice is the right answer, but we chose to do comprehensive genomic profiling. And want to show you what happened with him. So this was his genomic profile. And the first thing I want to point out is we now know why he didn't respond to the hedgehog inhibitor pathways. You know, the patch1 is the key alteration in that pathway. He had a patch1 alteration, but he didn't respond. Well, I think if you look at his genomic profile, it's pretty obvious why he didn't respond, because he had a whole lot of other alterations. He had a very high tumor mutation burden of 79, and he also had PD-L1 amplification, which we now know is a predictor of response. So remember, we treated him in October 2015, and we gave him nivolumab. This is his liver 2 months. This is his liver before nivolumab. This is 2 months later. He achieved a complete remission, and his response is now ongoing. In October, it will be 6 years. So this is something that we've never seen before, or in solid tumor oncology, maybe in testicular tumors, but otherwise not, is the ability, by matching patients to the right drugs, to get durable, complete remissions. I don't know when you call this a cure. I'm always hesitant, but I actually think he's cured. We're going on 6 years now. But I want to say, because everybody shows you their good results, that it's not all good. Love immunotherapy, but we also began to notice that there were a subset of patients that did poorly with immunotherapy, and not just poorly, but we felt that they were accelerated progression. And we've published on that. Institute Gustave Roussy published before we did, and now I think a lot of other groups have published on it. So I'm going to give you an example. This is a 44-year-old woman with triple-negative breast cancer. This is a tumor 2 months before therapy. She was getting radiation to the brain, so she wasn't receiving systemic therapy. So she had a little bit of tumor growth in the next 2 months, but not much. And then we put her on nivolumab. And this is what happens. Within 6 weeks, her tumor explodes. And she had an MDM2 amplification. And so we began to do a study at UCSD looking at what are the molecular alterations in these patients that have what appears to be an extreme acceleration in the progression of the disease. Sometimes the disease grew 35 to 40 times the rate that had been growing before. And this is what we noticed. We noticed 2 alterations. One, EGFR alterations, which for the most part seem to be associated with just plain old resistance to immunotherapy, but in about 20% of cases were associated with hyperprogression. And then MDM2 amplification, which seemed really almost always to be associated with hyperprogression. Now, one question that you could ask me is, so what is the mechanism here? And the answer is that I don't know, which is really frustrating. So this is a clinical observation, and we have some of our basic science colleagues trying to figure out why would this happen. The other thing I want to emphasize is that not every patient with MDM2 amplification is going to hyperprogress. Most of them will, in our experience, but not everyone. And furthermore, that there are patients that have this accelerated progression that have neither EGFR nor MDM2, so they have a different mechanism. But the important point here is that NGS can help define not only who to treat, but who not to treat. And that's probably as important as knowing who to treat. I also want to talk a little bit about really novel technologies. And here's a patient that I treated in January 2016 in our rare tumor clinic. She had a high-grade neuroendocrine cervical cancer. She was 49 years old. She had come in from Saudi Arabia in January 2016. She'd had surgery, chemotherapy, and radiation therapy, and she was really in bad shape. She had a huge abdominal tumor. She looked like she was 7 months pregnant, but it was all cancer. She had an impending bowel obstruction. She had partial kidney obstruction. Our urology consultant, who tends to be aggressive, didn't even want to put a stent in. He said she just needs to go to hospice. And frankly, if she lived in San Diego, she would have gone to hospice. But because she had come from so far away, we felt some sort of obligation to try and do something for her, which of course would be genomic profiling. But we really had a problem because we didn't think she would live more than a week or 10 days in the shape that she was in. And you know, we didn't have time to get a tissue biopsy and send that off for profiling because, you know, you put all that together and it can take 3 or 4 weeks. So we did a liquid biopsy on her, a blood biopsy. And here again, we have technology that honestly we could not have imagined back in 2012 or 2013. So by the time I left UCSD, we'd probably done about 6,000 patients. And a small subset were urine, most of them were blood, liquid biopsies. And so the technology is such that small amounts of DNA from the tumor are shed into the blood. And so we can look at the blood and know what's in the tumor. And this was what we found. She had profiling by Guardant. And, you know, when we've looked at a lot of profiles, most patients have 4 alterations, including the VUS, and she had about 20. So this had not yet been defined then, but we just made a guess that she had a high mutational burden and that she had hypermutated ctDNA. Today, we know that we can read tumor mutational burden off ctDNA, but this was January 2016, and this is what we saw in her blood. So we treated her with nivolumab. And this is her tumor before, this is her tumor afterwards. She achieved a complete remission, and I lost track of her after 3.5 years. She'd gone back to Saudi Arabia, but at 3.5 years, she was still in complete remission. So again, things that we've never seen before, using a simple blood test to give the right therapy to patient and a durable complete remission. After we treated her, we still wanted to know what was in her tissue. So we did a tissue biopsy, and the tissue biopsy really confirmed what we had assumed from the blood. She had 23 alterations. This is a Foundation Medicine panel. Usually we see about 3 or 4 alterations. She had a high tumor mutational burden, 53 mutations per megabase. And she had a mismatch repair gene defect and was microsatellite unstable. So that is the underlying biology by which she had this really remarkable response. So important point. Cutting edge technologies are needed for complicated cancers. And now we can do blood-based tumor mutational burden and microsatellite status. So that is stage 1. And now I want to bring you to stage 2. And what if every patient with metastatic disease is different? So now we go back to our snowflakes, and you see a bunch of snowflakes here. Each one has a unique crystal structure, and each one's crystal structure is very complex, and that's what we think about metastatic tumors. And so in order to try and articulate what we began to think about, I'm giving you this list of patients with breast cancer where we did Foundation Medicine testing, and we've done thousands of patients, but they don't fit on the slide. So I think I'm going to show you 2 patients in order to try and get the message that I want to get across. And so this patient has ERBB2 amplification, and so does this patient. And under classic oncology, which is the way I've always practiced and everybody else, this patient goes on the same regimen, these two patients, because they both have ERBB2 amplification. They go on a HER2-containing regimen. But the problem is, if you look at this, each of these patients, the rest of the alterations in their tumor are different from each other, and they have multiple additional alterations to the ERBB2 amplification. So if you think about what the science is telling us, it's saying these two patients don't belong on the same regimen, and one drug isn't going to cut it here because you have multiple problems here. You need a customized combination. That combination is going to be different patient to patient. And so instead of using a consistent drug between patients, even in the presence of different molecular profiles, which is the traditional way, we began to use a consistent strategy, which was molecular matching, but allowed different drugs for every patient. And yeah, so I'm not going to repeat that slide. So this is a drug-centric trial. It is the traditional trial. As stated during my introduction, I've led one of the largest clinical trials departments in the world, so I'm very well aware of how trials are conducted. You have three patients here. They have a commonality, which is an abnormality in gene A. So they all got drug regimen A, but the rest of their portfolio is different. So we don't think that that's ultimately the best thing for the patient. So patient 1 needs drug regimens A, B, and C combination, and patient 2 needs drug regimens A, E, and F. But that presents a problem because we've never done that in oncology. And if we have 300 drugs in oncology, if you want to do 3 drug combinations, that's 4.5 million combinations. We're never going to do all those clinical trials. And then you layer on top of it that you have to do 1 in each histology. That's just not happening. None of us are going to live that long. And so we really have to think about what the science is telling us to do, and we have to do a smarter and different type of trials. And then I want to point out to you that what I just talked about, which is patient-centric medicine, as physicians, we practice it every day. Oncology is actually the exception to the rule. And so I'll give you an example. If we have 2 patients with both have diabetes, but one has heart failure and depression, and the other has rheumatoid arthritis and COPD, as physicians, we have no problem. We never think twice about individualizing their therapy. The average patient walks into the oncology clinic because of comorbidities on 8 different drugs that have nothing to do with cancer, and all of those drugs are individualized to the patient's problem. So cancer, it turns out, is actually the exception to the rule, where we don't customize therapy. And I think that's a legacy of the chemotherapy era, where chemotherapy was very toxic and combining drugs could be, have very severe side effects. So there became this cultural prohibition against combining drugs ad hoc. But I think based on what the science is telling us, we need to get over that. So we created a trial called iPredict. Investigation of profile-related evidence, determining individualized cancer therapy. And the point in this trial was activated in February 2015, and the point was to give customized combinations. We also wanted to bring it earlier in the course of the disease, so we had a subset of newly diagnosed patients. But I'm going to start with the refractory patients. The output of that trial, the first output, was published in Nature Medicine in April 2019. And physicians chose the therapy, but the patient went through a molecular tumor board. And then the treating physician could decide what the therapy would be. And then we looked at patients that were highly matched versus patients that had a low matching score. So low matching did not mean unmatched. Some of those patients were unmatched, where they had a matching score of 0, but some of them simply had low matching. And so in its simplest form, if you have one alteration and you match that alteration, that would be 100% match. But if you have 10 alterations and you match only one of them, that's a 10% match. So in order to get a high matching score with 10 alterations, you have to match at least 5 of them. And that's where the individualized therapy came in. And this is what we found. So remember, these are refractory patients. The overall response rate was 45% in the highly matched patients. It was 16% in the low matched patients. The PFS was significantly longer in the highly matched patients. And then I want to point out the survival. At 2 years, the highly matched patients had an overall survival of over 60%, while the low or no matched patients had an overall survival of about 20%. So higher matching score translated into significantly better response rate, progression-free survival, and overall survival. This was not a randomized trial, but we've now repeated this several times, and really we get the same results. We had a very large paper that we published from our molecular tumor board with 715 patients, 429 of which were treated, exactly the same results. And the iPredict has a second arm that is almost accepted now in another journal. So there's another validation cohort as well. And I want to give you some examples of, what we were doing. So this is a 62-year-old man with poorly differentiated carcinoma of unknown primary. He has an unusual KRAS mutation, but it is activating and it is pathogenic. And we gave a MEK inhibitor, trametinib. He has an ARID1A splice site alteration, and with that we gave a PARP inhibitor because ARID1A can sensitize to PARP inhibitors. And you can see what's happened to his tumor. He had a progression-free survival that lasted 24 months. So nice response with individualized therapy. This is a patient of mine with 61-year-old man with metastatic anaplastic thyroid cancer. He had respiratory failure with intubation. He was on the ventilator in the ICU and was very religious family, and the wife was just really not willing to let him go. So we came to a compromise, and we said, we would try therapy, which was, a matched therapy, and if he didn't respond, she was willing to take that. So next generation sequencing showed that he had a BRAF mutation, and immune profiling showed that even though his tumor mutational burden was low, he was microsatellite stable, his TIL cells were low, his PD-L1 was high positive. So he had an extremely dramatic response. You can see his lungs here before. He was treated with emarufinib plus nivolumab. That you can see the improvement in the lungs. And elsewhere, got off the ventilator, actually was driving back and forth from Los Angeles to San Diego to see us, and had a great response that lasted for about 1.5 years. Unfortunately, relapsed after that. He did die. But I think this shows the power of, even in a very ill patient, of matching that patient to the right drugs. So I want to finish off by talking about the Worldwide Innovative Network for Personalized Cancer Medicine, which I've worked with since 2010, was founded by John Mendelsohn, who is the president of MD Anderson at the time. And the mission is global delivery of precision medicine. And the belief is that genomics, which is all I've talked about, is, or for the most part, is the tip of the iceberg, and we need to be looking at transcriptomics, proteomics, epigenetic changes, and so forth. And so WIN, as a start, has concentrated on transcriptomics. And this is the winter trial that we published in Nature Medicine. It was performed across 5 countries, Canada, U.S., France, Spain, and Israel. So again, international consortium, and to my knowledge, winter, and to my knowledge, it was the first precision medicine that included transcriptomics in addition to genomics for solid tumors. I enrolled 303 patients, 35% were treated, and about a third were treated on the basis of transcriptomics. So we found that in the clinic, transcriptomics could add to what genomics was telling us. And of course, the next iteration of this trial is combining transcriptomics and genomics. And then I want to mention one other type of new trial that I've been really involved in with leading, and that's a master observational trial. So this is a master registry of patients really across the nation where we want to get structured data. It's an observational trial, very similar to some of the observational trials, the master protocol that we did at UCSD, but this is on a national basis. And yeah, so just a couple of things to round this out. What about the host? I've talked a lot about the tumor. I don't think we've done enough about the host, and the host controls toxicity, response, and so forth. And this is a real person, and then you could ask, okay, so she made it to 100 years old as a chain smoker, pretty healthy in your face. Was this good luck, or was this germline genetics? And I don't know the answer, but I think it's germline genetics. And I think that we're coming very quickly to the point where we're going to do germline testing on every cancer patient. And then I want to finish off by saying that the genomic immunotherapy era will revolutionize not just cancer medicine, but I believe all of medicine. And I want to give you this example that I personally find fascinating. So we know that FGFR3 mutations are a hallmark of some tumors like bladder tumors, and we even have therapy that is approved, FGFR inhibitors, for this. So the very same tumor, the very same mutation, same gene, same locus, if in the germline, causes achondroplastic dwarfism. And this is fascinating for 2 reasons. One is, in the case of achondroplastic dwarfs, there is no increased incidence of cancer. So what is the mechanism that prevents, if you have this in the germline, there's some sort of compensatory mechanism, because if you get these FGFR3 mutations, let's say in your bladder, as a somatic mutation, that is associated with cancer. And then the other question, which I think is really interesting, is could we use these inhibitors to change the phenotypes of these babies? I don't know the answer to that, but the experiment has been tried in achondroplastic mice, and in achondroplastic mice, it works. So I think this opens the door for, we talk about repurposing drugs for cancer, but repurposing drugs from cancer to other conditions in medicine may also be possible. So I want to finish, and if anybody wants to contact me, leaving my Twitter handle and my Gmail address, and the Gmail is unusual. but people always ask me where I got it from. It stands for the Emperor of All Maladies, which is a bestseller that was written in 2010. And it's a phenomenal book, but I think it has one problem, won the Pulitzer Prize. It claims that cancer is too complicated and we're never going to be able to overcome it. And I think that's wrong. I will go back to where I started. In the 1990s, the conventional wisdom was that the human genome was too complicated and we would never be able to sequence it. And we sequenced it by 2003. So I'm hoping that this great book is wrong in one of its basic premises. And I'll finish there and happy to answer any questions.
Thank you so much for that great talk. It was wonderful. I'd like to ask Dr. Benny Carnero, who co-directs our cancer therapeutics program at the Cancer Center and also leads our early phase therapeutics effort at our institution, to moderate the questions. Benny?
Thank you, Wafik. Razelle, congratulations for the talk. Wonderful, as always. Maybe I'll start with one question and relates to your trial using transcriptomes to tailor therapy. We're all familiar with the concept of the mutational profile changing from tumors as they go to different metastatic sites, and it always intrigues to me is what's the impact on the microenvironment in different metastatic sites on the transcriptome profiling, and any efforts in terms of exploring those different profiles in different sites of disease. And the ultimate question is, are we going to ever be tailoring therapy to the type of metastatic site as opposed to only the histology?
I think that's a really good question. And so it's not just about the tumor, it's about the environment of the tumor. And in addition to that, one of the things that we did in the winter trial, which we found, is that looking at the transcriptomic in the tumor wasn't enough. We had to compare it to the normal tissue. And if we did not compare it to the normal tissue, the results were much less helpful. I absolutely think that transcriptomics will become a very important part of what we do, and that we're going to be looking at both tumor tissue and the microenvironment of that tissue and what it's telling us.
Thank you. If anyone has any questions, feel free to unmute your line or send secure chat. So I think we have a question from Attila.
Yeah, so the question is, your patient from Saudi Arabia's ctDNA panel list had fewer mutant gene signatures than the tumor biopsy NGS panel. Would this represent a challenge for ctDNA profiling since it misses many other gene mutations? Well, the answer is absolutely yes. The ctDNA panel list that we used back then had about 54 genes, while an NGS profile would have 200 to 300 genes in that era, probably about 200 genes. So first of all, the number of genes that were looked at were fewer. The technology is getting better. A lot of ctDNA panels now have 70 or 80 genes, and there are now panels that we have 300 plus genes. So I think the technology is improving. Number two is that you're looking at extremely small amounts of DNA that are shed from the tumor into the blood. So that is absolutely going to be a challenge. My strong belief is that for the foreseeable future, we need both the blood biopsy and the tissue biopsy. They are complementary, and we do both of them. That just one of them, especially at the time of diagnosis, is not enough. You really need both. In the patient that I presented, we started with the liquid biopsy because we didn't have time. She wasn't going to live long enough to get the tissue biopsy. But in general, we start with both a tissue biopsy and a blood biopsy.
Okay. And I think we have another question from Anne-Louis about how do you approach tumor heterogeneity in designing precision medicine regimens?
Yeah, so that is actually a key question. And of course, we don't know the entire landscape of heterogeneity. We're not biopsying every tumor, although we have some ideas because we're doing liquid biopsies. So what we try to do is we put together all the data from all the biopsies that we have, all the data that we have from the liquid, the tissue, and if we've done immunomomics, if we've done genomics and transcriptomics, and then we try to prioritize what we think are the most important drivers and how many of them can we hit with remembering that you probably can't give a patient more than 3 or 4 drugs without running into trouble. And we're sort of, I think, breaking the glass and giving customized combinations. But 3 or 4 drugs, 3 drugs is usually the limit, 4 if you're really brave. So the science isn't perfect. We know some genetic defects are more likely to be drivers, like, for instance, fusions, we believe are more likely to be drivers. But sometimes we just can't hit everything, and sometimes we're not sure which is more important.
Hi. Sorry. Can I ask a question?
Sure.
Yeah, thank you. So first of all, it's a great talk. And my question is, if a patient which has got the immunotherapy or after the complete cancer treatment, but if in case the circulating tumor cells in the blood streams are present, did you notice any patient who come back after the immunotherapy with different type of mutational burden?
After immunotherapy in patients who have a non-complete remission, right? And you're asking about tumor mutational burden, or you're asking about new gene alterations?
I'm asking about new genes alteration.
Okay, so yes, we've seen new gene alterations emerge, and I don't think we've done enough in that space, but interestingly enough, one of the gene alterations that we've seen emerge is alterations in EGFR. And I mentioned before that EGFR alterations are often an indicator of resistance to immunotherapy, de novo resistance, and they may also predict for hyperprogression. But we have absolutely seen new gene alterations emerge, and the ones that we think are pretty interesting are the EGFR ones, because it makes sense for us biologically. Now, if the patient attains a complete remission, we're just not going to see that. The liquid biopsies are going to be negative. But if the patient has a partial remission, we can often predict a few months before the relapse that the patient is going to relapse, because we begin to see these emerging alterations that were not there before.
Okay, thank you. Another question is how frequently the patient is getting these immunotherapy doses, and what's the range of the dose they are getting?
With the immunotherapies?
Yes.
We generally give the FDA-approved doses with the immunotherapies, and we will occasionally, which a lot of people don't do, but we will occasionally reduce the dose if the patient has toxicity, but we still want to continue the regimen, so we may cut the dose in half. A lot of people just stop the drug or obviously give other drugs, anti-TNF or other drugs to ameliorate the side effects. But if we're running into a problem, we may also decrease the dose of the immunotherapy. And then one of the questions is always, if the patient attains a complete remission, how long do you continue on the therapy? I don't know the answer to that. We do it as an individual judgment, but I don't think anybody really knows the answer.
Thank you.
I think I have one question from Fik. What would you say to the skeptics about whether we should sequence most patients based on how many patients are being helped? What will it take for the practice of oncology to practice in the way that you have been, to help even more very sick patients?
So in our program, about 50% of patients were getting matched to therapy. 40–50%. I know that in a lot of other studies, it's 5 to 10%. I personally think there's several barriers, and this is why only small numbers of patients are being helped. The first barrier or challenge is that whoever's leading the molecular tumor boards and stuff has to be really knowledgeable in genomics, and that doesn't always happen. The second really barrier is operational, and that's because people can't get the drugs. And they either don't have the clinical trial availability. So while I was at MD Anderson, the way we got drugs was clinical trials. MD Anderson puts 10,000 patients on trials a year, and we had the largest clinical trials program in the world, or phase 1, where we put 1,300 patients on therapeutic trials a year. So it was mainly clinical trial focused. UCSD, when I was at UCSD, much fewer clinical trials. So we had a clinical trial navigator there, but we really needed to be able to get off-label use of drugs, and that was a key to being able to do this. And so what we did is we hired a magician, and so this is a joke, but we hired a medication acquisition specialist, and I call her a magician because she's really outstanding. She could get 95% of the drugs that we needed, and there are many mechanisms to help patients get medication in the United States. There is really, it's a very rich resource of mechanisms. The problem is that the physician in the clinic with 25 patients cannot navigate that. Impossible. So we simply solved that problem by hiring a person that was extremely dedicated and figured out how to navigate the whole thing. So the physician would just call, she would come to molecular tumor board, she would call, or the physician would call her or email her and say, these are the drugs I need. And she would say, yeah, okay, 2 weeks, it'll take me. She knew everything. She knew all the systems. The physician didn't have to do anything. So I think part of it is operationalizing a new paradigm. If you have a new paradigm, you have to make it work. And the challenges are not about NGS. The challenges are often practical. Like, you know what drug you want to give the patient, but you can't get that drug, and the drug costs $10,000 a month. Those are the practical challenges that need to be solved.
So Razelle, just a follow-up to that, with those challenges, how are you trying to deliver precision medicine in other countries where even genomic profile is limited, and as you plan the trials to be globally
Well, you would be
active?
surprised that there are physicians that are extraordinarily innovative, a lot who I met through Twitter, which is like, can be poisonous, but can also be amazing if you stick to medicine. There are innovative people all over the world which are doing like fantastic things. And so I'm going to just give you two examples. I met a physician from Turkey that we now collaborate with, and he told me some of the patients he was treating, and he's like doing advanced precision medicine. And so, you know, we had a Zoom call because he wanted to do a project together. So I said, so, you know, what you're doing is amazing. I love it. How are you getting the drugs? Like, I don't know anything about Turkey. And so he said, well, in Turkey, and this would be totally illegal in the United States, patients who die or who progress bring back the rest of their drug to the hospital. And he's allowed to use that drug supply, and it's free. And so that's how he gets them the drugs. And, you know, I don't want to go through it, but I have a physician in India that I've spoken to. They have she has a completely different way, but she also gets the drugs. And I have collaborators in Israel, and they get the drugs. In each of these countries, the way to get the drugs is different. And frankly, in the United States, we could never recycle the drugs. You'd lose your license if you did that. Somebody brings back the drug and you give it to somebody else. That can't be done. But we have a different mechanism here. And there may be some countries where there is no mechanism. But what I've been amazed at is individual, really smart, innovative physicians have figured out mechanisms in countries that I would never dream. You know, Turkey, India, Israel, that I just would not dream. And the mechanisms are different.
So just as a follow-up to that,
question as well.
At Brown University, there is a startup started by a couple of Brown undergraduates called MediCircle, whose goal is to do exactly that in the United States, and it's taking a lot of traction, and it's exciting. We'll have to see how it goes with it.
I would love to learn more about it. I would love to learn more.
I do have a quick question, if you don't mind.
Sure.
Thank you so much for a great talk. I have a question about the tumor heterogeneity and the genetic heterogeneity between the main tumor and then the metastasis. Do you feel that moving forward, it will be maybe beneficial to include the liquid biopsies to have a full spectrum, you know, of the mutational burden of the especially metastatic patients that sometimes may have different clones in metastatic sites? Just how to get really comprehensive insight into the complex genetic complexity.
So we test tissue and liquid simultaneously, and that's at diagnosis. And then if the patient relapses, what we want to do, not that we can do it for everybody, but we want to biopsy the growing lesion because, like, maybe not every lesion is growing, and we want to do a liquid biopsy.
Okay.
So we definitely do both, and we believe that the results are complementary. They're not identical. They're not supposed to be identical, but they're complementary tests.
Okay, great. Thank you.
Thinking in respect of the time, maybe we can go through one more question, so if you have a few minutes. It's from Attila asking, since sequencing becomes so cheap, do you foresee in the near future many consented patients, people with DNA will be sequenced for the screening of germline mutations, provided that identity will be protected and will not be used against them by employers, insurance companies? I think this also relates to the large, a lot of genomic information in companies like your genes and
yes.
publicly available.
So if you're talking about cancer patients, I think that in the very near future, every cancer patient is going to have germline sequencing. I've seen many patients whose tumors apparently didn't read the books. And I saw a patient, a 73-year-old with P53 mutation. It was his first cancer, no family history of cancer. As part of a research project, he had germline sequencing. He has leishmaniasis. And that was really important for him to find out for his children. But he met no criteria. And I think that there will be data coming out that at least 10 to 20%, at least, of cancer patients have a germline alteration. So I do believe that germline will become de facto for every cancer patient. Will everybody be sequenced? Every human being? Maybe. So there's a couple of things that I want to just say about protection. There is a law in the United States that the germline sequencing cannot be used against you. Now, I don't know if anybody believes that that law really protects you, but, because of course, now you're going to sue some big insurance company, and good luck to you. But there are laws that supposedly protect you against it being used against you by employers or insurance companies. And then the last thing I want to say is somebody told me something really interesting about a decade ago, and it was somebody from insurance. And they said, people are afraid to do germline sequencing because they won't be able to buy medical insurance. And he says, as an insurance adjuster, I'm more afraid of the opposite. People are going to do germline sequencing, and most people are going to find out that they're going to live a normal lifespan. They're not going to want to buy insurance. He said, we live off healthy people buying insurance because they're afraid that they're going to get sick, when in fact, if they knew their germline, they may realize that they're not going to get sick. So I just wanted to say that there's different viewpoints, and sometimes a little more complex and nuanced than we think about it.
Thank you, Razelle, for a wonderful talk and discussion. I think, in respect to everyone's time, we'll need to wrap up.
Well, thank you for having me, and feel free to reach out if there's any other questions. But really, special thanks to all of you for the invitation and the great questions, and I hope we can find some way to work together. You take care.
Thank you. Bye.